The work that I'm going to tell you about today was initiated in our lab by Allison Hickson, an MD PhD student, and then carried on subsequently by Michael Rudy and Josh Frost. And we're grateful for the organizers of the symposium for inviting us and for our various funding sources. We began this project, meeting Allison, by just looking to see whether or not if we took isolates of enterovirus D68 obtained in the 2014 outbreak, whether or not if they were put intracerebrally into outbred mice, they would produce neurologic disease consistent with AFM. And all I want you to see from this slide here is that a number of 2014 isolates belonging to a variety of clades, shown here as B1, B2, and D, did produce neurologic disease in mice after intracerebral inoculation. And this was something that was different from what we saw with the prototype Furman and Rin strains inoculated into these mice by the same route. I'd also point out there's one strain there that you can see, the California 144231, that hasn't produced neurologic disease by this route in mice. So it is not a feature of all strains. In Swiss Webster mice, just using the Illinois 18952 strain as a prototype, we found that motor impairment, meaning the mouse equivalent of flaccid myelitis, was dose dependent, so higher doses of virus resulted in more severe neurologic impairment and more rapid onset. We also noted that the route of inoculation could determine the frequency and prevalence of neurologic disease. So intramuscular inoculation, for example, is an even more efficient route in Swiss Webster mice than intracerebral. And routes such as intranasal or intraperitoneal inoculation do produce identical paralytic disease, but much less commonly than either the IC or IM route. And in a sense, that's almost what we've seen in the human model, where respiratory infections overall are, of course, far more prevalent than AFM. The Swiss Webster mice also replicate the age dependence of enterovirus D68. And here you can just see a series of curves, with the highest curve shown there, implying more impaired motor performance by mice in mice injected at day 3 of age. And by the time you get down to about 2 weeks or 3 weeks of age, we no longer in Swiss Webster mice see significant neurologic disease. We just showed the age-related susceptibility of the mice to paralysis. If you just show the video, this is just to give you a little example of what one of these paralytic mice looks like. It just takes a second or two. And you can see this poor fellow is dragging both of his hind limbs behind him. We also get individual limb paralysis as well as this paraplegic pattern. If you take the spinal cords out of those mice, as shown on the next slide, you'll see that we get infectious virus and viral RNA peaking at about 6 days after inoculation in the mice. And if you look at the spinal cords of these animals, you find that there is loss of motor neurons, here marked by a choline-acetyltransferase staining, corresponding beautifully to the side and part of the spinal cord innervating the paralyzed muscles. So if you look on the right-hand side of that slide where the vertical arrow is, you see almost no choline-acetyltransferase positive neurons, whereas on the preserved right side, they continue to exist. And we can find enterovirus D68 antigen in the residual neurons, or if we go a little earlier, in more neurons. Here's just a close-up showing this at a little higher power of a enterovirus D68 positive neuron seen there in the bottom figure in the spinal cord. If you do electron microscopy of these spinal cord sections, you can see particles corresponding morphologically to what we would expect for enterovirus virions within cells, including neurons. So you see at 11,000, 49,000, and 68,000 98,000 power, those particles, and just a reference in the lower right-hand corner from the CDC of what enterovirus D68 particles in cells should look like. Although I focused in on the fact that the virus infects the neurons here and kills those cells, I don't want to leave you with the impression that there's no associated immune response. And here you can just see from approximately 12 days after infection, a neuronophagia with an infiltration of CD3 positive cells occurring in one of those spinal cord sections. If we use a system very similar to what Amy described, in either spinal cord slice cultures ex vivo or in the spinal cords of mice, we can find elevations in a number of pro-inflammatory cytokines. Now, I will say if we look at the temporal profile of both of these things, they seem to occur after both the peak of viral replication, which I showed you earlier, and after the onset of the neurologic disease. We've used this model system to test a variety of therapeutics. We began with some studies using human IVIG. Human IVIG contains high titers, virtually all lots that we and others have tested of neutralizing antibodies against enterovirus D68. And here you can see that in a time-dependent manner, compared to PBS, which of course has no effect, that if you administer human IVIG immediately following viral inoculation, such as day 0 or slight delays like day 1 or 2, you can virtually prevent the development of neurologic disease. And even by day 4, when virus has already reached the nervous system and we have had onset of paralytic signs, you can have a similar effect. I didn't show a day 6 curve, but as you might imagine, it sits between the day 4 curve and PBS. There's a great correlation between capacity to protect by IVIG and other agents and both the ability to reduce viral titer in the muscle and prevent virus from reaching the spinal cord. So you can see here in the right-hand IVIG columns in both of the two panels, B and C, the striking difference in the amount of virus in muscle on the left and spinal cord on the right compared to controls. We've also extended this by using monoclonal antibodies in collaboration with Zab Bio. These monoclonal antibodies are chimeric human mouse antibodies and basically have the same binding characteristics as Zhang and Al's antibodies as described in Nature Microbiology that Richard, I think, alluded to before. Zhang and colleagues in a different model were able to show that 2 of these antibodies, designated 11G1 and 15C5, had protective capacity. When we put these antibodies into mice, in our model that I just described to you before, we also found that the 15C5 antibody shown here in red, given a day after viral challenge, was able to protect mice against development of paralytic disease. In our hands, the 11G1 antibody was not effective, nor was an isotype matched or equivalent anti-herpes virus antibody used as a control. We also used a model that would resemble something closer to a therapeutic model, which is to say we waited until animals showed clinical evidence of neurologic disease, meaning a paralysis score of greater than or equal to 2, which was detectable weakness in one or more limbs. And we're even able to show that something closer to a treatment paradigm was still able to demonstrate protective capacity with this 15C5 antibodies. Not everything is effective in therapy, and so of course when there was much interest in fluoxetine as a potential antiviral agent, we tested that drug in mice, and I'll just quickly say it neither was effective in terms of impacting paralysis nor influencing viral titer in either muscle or spinal cord. And more recently, we've been collaborating with Jun Wang at the University of Arizona. Many of you are aware that he had a paper in the Journal of Virology, which I've just shown up there just a little over a year ago, indicating that the hepatitis drug telaprevir was potentially efficacious in inhibiting enterovirus D68 replication in vitro. And we've tested this at a variety of doses now in mice and report that there is a dose-dependent effect in mouse models. Now, I will say our preliminary studies, which we started just before COVID kind of put us in lockdown, that we just are emerging from now, suggested that unlike what I showed you with IVIG, if you delay onset of treatment with tolapivir, we don't clearly see therapeutic effects. And we're currently now looking at combinational therapies with the use of an effective antiviral, potentially tolapivir, eliminate, for example, the potentially deleterious effect of steroids. Next slide. And I think this is pretty much the last slide coming up. It was just to remind you that when corticosteroids are used alone in our mouse model, that we not only see enhanced lethality, but among survivors, enhanced motor scores, meaning worsened disease. And so again, because there is some interest in using immunomodulatory agents other than antibodies, it would be nice if those could potentially be combined with an antiviral to see if they had less deleterious effects. Next slide, or I think that may be the last one, or you can go ahead. This just shows that the corticosteroid therapy is associated with higher titers in both muscle and spinal cord. I guess I should remind people at this point that that concludes my talk. Again, apologies for all the little technical glitches. Remind you that for the Q&A period, and I think we'll hear more from Carlos about that, that you have to go to your sessions window in the hop-in format. And thanks again, and sorry I sort of had to rush through that, and apologies for the technical issues. So I'll see everybody in the session room. Thank you.